Hey, 123 A&P, my friends. I'm inspecting some of my voice threads here, and I've found that the cellular level of organization, part 3, membranes and transport, a little bit boogered up here. So I don't know if my giving students the full product, my username and password, somebody's taking liberties, or something inadvertent, or I screwed up, whatever. Hey, check yourself before you riggedy wreck yourself. Just view these things. Please don't edit them so that other students will be able to access the content. So let's look at part 3, membrane transport. We know that the membrane is a phospholipid bilayer, right? We can see that there are these phosphate heads and the little fatty acid tails, and to me it looks like a little bit of an ice cream sandwich, all right, with all the fatty, creamy goodness in the middle, and then the two ends of the bread on each side. And I like to let it melt just a little bit before I sink my teeth into it, chase after the ice cream lady, you know what I'm saying, or the ice cream man, whatever. You can see we have these various membrane proteins where we talked about. They extend partly or fully through the membrane itself, and they've got some extensions, things that will come out of the membrane like antennas, and they kind of feel for the external cellular environment, what's going on. They're means of communication. They will sense different molecules in the chemical environment, test the waters, so to speak, in the interstitial fluid, that is the fluid between cells, and then will allow us to interact quite a bit. Sometimes we'll get substances that will pass through these channel proteins freely. Sometimes we'll need a little doorman to say, hey, yeah, okay, we'll let you in, and the information or that chemistry will float through. And other substances are more readily absorbed through the membrane than others. We call that barrier permeability. All right. So here we can see in orange we've got some substances that are approaching the cell. Maybe they'll be able to get through the phospholipid bilayer without any problem, through osmosis. Maybe they'll just be incorporated into the cellular membrane. Some things are going to be too large or too polar to get through the cell membrane, and other things they can get through. They can gain access to the cell, but they're going to need somebody to give the okay. I use a little doorman analogy, like security, you know what I'm saying? Or try to get in a club, yeah. So diffusion is a passive mode of transport into the cell, all right? So it doesn't require any energy. The energy of the energy currency of the cell being adenosine triphosphate, or ATP. That is what gets all the work done in and around the cell and in biological systems. Yes, yes? All right, so here we can see on the left, we've got a solute, let's say it's in an aqueous environment like the body exists in, and then the particles are going to start to pass across the membrane, and then eventually there's going to be an equilibrium where particles or solutes are free to flow at will across the membrane. That is a passive process called diffusion. Here's a little example of some substances that are able to gain access to a cell with very little resistance. So we've got nonpolar molecules, which means that these molecules are not incredibly reactive chemically, all right? So they can gain access to the cell. Fatty acids can get in. Fat-soluble vitamins such as A, D, E, and K are able to incorporate into the cell membrane because it's got that lipid component. Steroids are also made of fat, and so they'll be able to gain access. Oxygen, CO2, and water will also be able to diffuse across the membrane. Larger substances, okay, or reactive ions such as sodium, potassium, calcium, hydrogen ions, reactive molecules, large molecules like amino acids, and imagine those beads on a string. Glucose is pretty big. All right, nucleic acids. They're going to have problems. So either they're going to need some facilitation, some help getting across the membrane, or they won't be able to make it through at all. Here's a look at osmosis. Osmosis is a passive process where solutes flow in water, dictated by how much pressure is in the environment, what the composition of the fluid, the aqueous fluid, concentration of solutes. Imagine salt being dissolved in warm water. It's going to naturally spread and flow until those molecules all reach equilibrium. All right? And this little dividing line down the center, of course, is the membrane. Tonicity, okay, when we talk about tonicity, that's how much of a dissolved substance exists in an aqueous solution. So again, go back to sodium. Sodium chloride. You put it in water, it disassociates, whatever. You gargle, you drink Gatorade. Sodium is one of the primary constituents of our cellular environment. In fact, in the extracellular environment, or the fluid between the cells, we call that the interstitial fluid. Sodium is a primary ion. Now, inside the cell, this red blood cell, for instance, there is a lot of potassium. Potassium likes to live within cells, and so there's this cool gizmo called the sodium-potassium pump that strikes the balance between the sodium, that keeps that ratio of sodium outside the cell and potassium within the cell. It pumps so we can maintain that proper balance of sodium and potassium. Hence the verbiage, sodium-potassium pump. All right. So what happens if we take a red blood cell and we drop it in a hypotonic solution, which means that there's a lot of fluid and not a lot of solutes? What happens is that means that on this image on the left, you've got fluid inside the cell, primarily water. All right. In the extracellular, outside that cell, that environment, we've got a lot of water in that extracellular environment. So what happens is there's more tonicity, there's more stuff, solutes within the cell than there are in the extracellular environment. What happens then is, in order to strike the balance of osmosis, the fluid will go from the extracellular environment and it will move into the red blood cell in this case, and it will blow up. You can see that that RBC is expanding as it tries to maintain tonicity, a tonic equilibrium. So the cell will lyse or cut, blow up. Boom. Whereas in an isotonic solution in the center, all right, you've got a solution that's amenable, that's capable of maintaining life. So we've got just the right amount of solutes, sodium, potassium, chloride, magnesium, that are dissolved in the solution to water. The ratio is good. And so you see the little red blood cell looks like a raft that you would maybe sit on and go around a lazy river or something like that, right? That is a healthy red blood cell. That's the way it should be. If we go to the right image, we've got a hypertonic solution. So the red blood cell is bathed in a solution that is very salty. It's got a lot of minerals dissolved in the aqueous solution. So what happens then is the fluid from within the red blood cell, it tends to migrate out in efforts to maintain the equilibrium. So then it shrivels up, and it's called crenation. It dies. So we want it right in the middle. An isotonic solution is what's going to help us maintain homeostasis. Carrier-mediated transport, okay? So talked about passive versus active transport. Passive transport requires no authority, no invitation to get into the cell, gain access to it. But carrier-mediated transport means that there's a doorman or somebody on security standing at the gates of these channel proteins within the cell membrane. And when a molecule comes up and says, knock, knock, I want to come in, that doorman, the security, says, hey, all right, you can come in. You can come in, you can come in. But, hey, we're full. The dance floor is full. We've met capacity. We're not letting you in. Or you're suspicious or whatever. So the carrier protein just escorts the substance within the cell. This would be carrier-mediated transport. That's the doorman. Facilitated diffusion. All right. This is also going to require a doorman, some sort of authorization that is going to allow the substance into the cell. Facilitated diffusion is going to require some energy or an escort. And of course, active transport is going to require ATP. So the energy currency of the cell will have to bind and say, okay, well, we're going to let you in, but it's going to take some resources. So in that case, you pay the doorman in order to get into the cell. And a good example of active transport occurs with, as you see down bottom there, the sodium-potassium pump. Cool things about the cell membrane, we can, if there's something that we like in the extracellular environment, let's say we're in the cell, if we see something that's desirable, something that we need within our cell, we need some of that, you can actually, like a amoeba or something, you can pinch off part of the, like create a little mouth as you see this vesicle forming there. You can pinch off part of the membrane and swallow up that whatever that substance is, and then incorporate it into a vesicle, and then the vesicle will migrate into the center of the cell, drop off the supplies, and then go back for more. And there are different types of this. They call it pinocytosis, basically pinching off of the cell. Pretty neat how alive these cells are. We think of them as like these little structures in there that are just, okay, this is my skin cell. This is my cardiac muscle cell. This is my liver cell. They're functional, man. They're living a sci-fi world in there. So here's endocytosis or pinocytosis. Think about in terms of the immune system, you might have heard of the term phagocytosis. That means to eat. So a phagosome is like where an extension of the membrane is going to swallow up a structure, like a Pac-Man or Ms. Pac-Man, and bring it into the cellular environment. So that's pretty neat. Exocytosis, let's say we got to take the trash out. We've got some cellular functions happening. We're doing work inside the cell. As a result, we've got to take the trash out. We can't keep this substance within the cell, so we're going to basically puke it up and out into the extracellular environment. So endo means in and exo means out. Look at those root words, speaking the little Latin there. And that's the end of this this lecture here. It's pretty short, just talking about membrane permeability. Just know that in closing, there are some substances that we need in the cell and some substances that we need to get out of the cell. And these cells are working. They're like a bunch of cars that are idling in a parking lot. All right? They're doing work. They're producing. They've got various components called the organelles within the cell. They're enclosed by a membrane that seals them off from the outside world. But some things we can let in, and we need those substances within the cell. And sometimes things can just float in passively, requiring no energy, no assistance, no authorization from a doorman. And then sometimes substances are going to take some energy to get in. And other times we might need to get rid of some stuff. Think of the exhaust that flows out of our tailpipes as we're sitting there in that parking lot idling. In that case, we would do an exocytosis or something, right? All right, I just knocked out this voiceover, so this should be nice and fresh for you folks. Please enjoy.